Bom dia, boa tarde, neste programa Nanotecnologia do Avesso. Hoje, no seu número 362. Fazemos este programa para o público não especialista, porque é este público que paga as pesquisas sobre nanotecnologia porque é aqui que nós podemos discutir os impactos da nanotecnologia na economia, na sociedade, na ética, no direito, na saúde, coisa que não são discutidas aonde se faz nanotecnologia no Brasil. Por fim, fazemos este programa porque acreditamos que ser livre é ser bem informado e a divulgação científica contribui, colabora para o aprofundamento da democracia e da cidadania no Brasil. Hoje, conforme tínhamos anunciado, estamos conectados com a professora diretamente da Universidade de Manchester, Lynda, de pesquisa na área de nanotecnologia e saúde que ela vem desenvolvendo. Ok, Lynda, could you please introduce yourself?
Yes, I'm Dr. Lynda Harris. I'm a lecturer in pharmaceutics at the University of Manchester in the UK.
Okay, you can.
I didn't hear that clearly.
Yes, the first slide is in the screen.
Okay, thank you. Today I'm going to tell you about some of my research developing targeted liposomes for targeted delivery of drugs to the placenta. My interest has been in placental function and placental growth for a number of years, and more recently I've moved into novel nanotechnologies for targeted delivery of drugs to the placenta. So if we could go on to the next slide, please.
Okay, is in the screen.
Yeah. So during this presentation, I'm going to tell you some of the reasons why we might need to enhance placental function in pregnancy. I'm going to tell you how we might be able to achieve this and what kind of drug compounds we could use to do this. And I'm going to tell you about our ongoing research to develop different kinds of nanocarriers for targeted drug delivery. And then I'll finish by telling you a little bit about some of the nanoscale therapeutics that we're also developing, this time to enhance intrinsic growth signaling rather than deliver an exogenous growth signal. Next slide, please.
Okay.
So why am I interested in pregnancy? Well, almost 10% of pregnancies are complicated by various different conditions, and these can include preeclampsia, where the mother's blood pressure becomes dangerously high and her kidneys start to fail, fetal growth restriction, where the baby is born too small, macrosomia, where the baby is born too large, which typically occurs in mothers that have pregnancy-associated diabetes, and preterm birth, where the baby arrives too soon. So all of these complications have serious short-term consequences. If you could get to the next image, please. These include neonatal intensive care, risk of fetal death or disability, but also these complications have long-term effects on health in adult life. So if you get at the final image on this slide, you'll see that if you're born too small or too early, there's an elevated risk of various complications in later life, such as hypertension, heart disease, type 2 diabetes, and things like osteoporosis and schizophrenia. So if we can improve fetal growth in the womb and optimize fetal health at birth, then we are also optimizing health in later from our research is many of these pregnancy complications are caused by a placenta that doesn't work properly. So the placenta, or afterbirth, is the organ that transmits all the nutrients and oxygen to the developing fetus. And if it's too small, or if it's not developed properly, it can't transmit enough nutrients and enough fetus, so the baby's growth is impaired. What we know from our work with animal models is that if we can improve placental function, we can alleviate some of the maternal symptoms of pregnancy complications, and we can improve fetal outcomes and fetal growth. So by indirectly treating the placenta, we can improve fetal growth. If you could get the next piece of text up, there's various different drugs and drug classes that have been used in animal models to improve placental function. Growth factors, such as insulin like growth factor 1 and 2, can improve placental cell growth and survival. Antioxidants can reduce some of the oxidative stress that builds up when a placenta is not working properly. And vasodilators can improve uteroplacental blood flow, so you get more oxygen delivered to the placenta. And in various rodent models and larger animal models such as the sheep, these different interventions can all improve fetal growth. If you could get the last text up on the slide. The main problem now is we've identified potential drugs to be used in pregnancy, but the problem is it's not safe to administer them in humans, essentially, because there's concerns about detrimental side effects for the fetus and for the mother. So what would be really good if we could develop a way of selectively targeting drugs to the placenta, because that would minimize the risk of adverse fetal side effects. So I was reading various papers about targeted drug delivery in the cancer field and discovered that this was potentially possible. You could go on to the next slide, please.
Yes, is in the screen.
So maybe about 10 to 15 years ago, technology was started to be developed primarily from the Ruslati lab at University of California, Santa Barbara, where he was looking at ways of selectively delivering drugs to tumours to reduce the off-target side effects you get when you administer chemotherapies to humans. And what he did was he identified epitopes that were present on tumour cells and tumour vasculature that were not present anywhere else in the body. And he was able to identify tumour-specific homing peptides that bound to those epitopes. And that's how he created his targeted drug delivery systems. So if you could get up the first image, he used phage display using phage libraries to identify these tumour-specific peptides. And with a phage library, this is engineered so each virus particle expresses a single random peptide on its surface, but within the library, 10 billion different peptide sequences are represented, so it's possible to screen 10 billion random sequences in a single experiment. So if you could get the next image or next two images up, what you do is you take a small volume of this bacteriophage library and inject it into a mouse. And what you find is the random peptide sequences that selectively bind to the heart will accumulate there. Some of those random sequences will selectively accumulate in the kidney, others in the liver, others in tumours. So whichever tissue you're interested in, you need to dissect that tissue out of the mouse, grow up the viruses that are bound there, and then if you sequence those viruses, that tells you the sequence of the randomly expressed peptide on the surface of that virus, and that's your tissue-specific homing peptide. So I was lucky enough to spend a year working in the Ruslati lab, and I repeated his experiments. But instead of using tumour-bearing mice, I used pregnant mice, and I dissected out the placentas and sequenced the phage that preferentially accumulated there, and then I was able to discover a number of placental-specific homing peptide sequences. And once you have those sequences, you're then able to attach them to different kinds of nanoparticles to create your targeted drug delivery system. Next slide, please. So before I go any further, I just want to tell you a little bit about the structure of the placenta, so you can understand some of the images that are following. So looking at all of the images The first one, you have a human placenta here, and you see it's intimately associated with the wall of the uterus. It's very adherent. Early in gestation, it grows very rapidly. It has the appearance of seaweed with lots of finger-like projections called villi. And early in gestation, the placenta grows much more rapidly than the fetus, so it can provide a large surface area for nutrient exchange and support the growth of that developing baby. By term, at the end of pregnancy at 40 weeks, it's about the size of the dinner plate. It's not growing anymore, but it has various functions. It acts as a barrier, it acts to transport nutrients and remove waste from the fetus. We've also added a picture of a mouse placenta there for comparison. This is a mouse placenta and a mouse fetus that you can see in the image. It's taken at embryonic day 15.5, which is the equivalent to probably about 30 to 35 weeks of human pregnancy. Mice tend to have gestations between about 18 and 21 days. So if we go on to the next slide, please.
Yes.
So just to show you a little bit more about placental homing peptides, the images on the left-hand side of the screen are human placental tissue. So this is a section through first trimester, so early pregnancy, human placenta. And you can see the outside layer, there's a bilayer of cells, and they're called trophoblast. And they are the main placental cell type responsible for taking in the nutrients and excreting the waste. So if we wanted to deliver a drug to the placenta, this is exactly the cell type that we want to target. Underneath the trophoblast, there's a core of stromal fibroblasts and blood vessels. So if substances penetrate the trophoblast, they can move through the stroma and into the blood vessels and through the umbilical cord to the fetus. The image below shows some explants of human first trimester placenta that have been incubated with one of my placental homing peptides. So this is abbreviated as IRGD. The actual sequence is a cyclic 9 amino acid sequence. It has cell penetrating properties, so it's termed internalizing IRGD, but we've abbreviated that to IRGD, that's why I have that name. And it has a fluorophore, carboxyfluorosine, abbreviated to FAM, attached to it, so it emits a green fluorescence. And as you can see on the images at the bottom, after incubating the placental tissue with this fluorescent homing peptide, it accumulates in that outer trophoblast layer, which is exactly where we would want to deliver the drugs to. On the right-hand side, you have some images of the mouse placenta. At the top, you have a section through the mouse placenta, and you can see that it's made up of two sections. So you have the labyrinth, which is the bottom half, which is a network of blood vessels that transports the nutrients to the fetus. That's the fetal-facing side. And then the top part is the maternal-facing side. You have the uterine spiral arteries that feed blood into the top of the placenta. The band in the middle is referred to as the junctional zone, which has endocrine properties. It secretes hormones. So although the structure of the mouse placenta is slightly different, the functions obviously are common. In the bottom picture, you can see a mouse that we've injected with our fluorescent IRGD peptide, and you can see the green fluorescence of the peptide accumulating in that labyrinth layer, the area of nutrient exchange, on the two images on the right, but also it binds to the endothelium of those spiral arteries that feed blood into the top of the placenta. So using these peptide sequences, we can target both the placental vasculature and then the region of nutrient exchange. If you go on to the next slide, please. So once we've identified our sequences, what we wanted to prove is if we attach these peptides to a nanoparticle of choice, could we get them to selectively accumulate in the placenta? I was undertaking this work in Professor Rosati's lab at the time, and they had some iron oxide nanoworms available that they used for tumour imaging. So these are strings of iron oxide nanoparticles about 180 nanometers in length, and these could be decorated with control peptides or targeting peptides. So at the images at the bottom of the screen, you can see placenta that have been injected with these peptide-decorated nanoworms. And in these images, we're using the CGKRK sequence, which I also showed targeted the placenta selectively. You can see in the first image that we've got really nice staining again in the labyrinth area, and also in the first and second images, a nice accumulation in those blood vessels that feed into the placenta. The image on the right is a picture of a mouse placenta that was injected with peptide-decorated nanoparticles, but we used a control peptide that time instead of a targeting peptide. So you can see that there is no accumulation of the particles in the placenta. So this tells us that our targeting peptides do work and can courier a cargo selectively to the placental tissue. During these experiments, we did screen all the major maternal organs and the fetal tissues as well to make sure there was no fetal transfer. We saw transient accumulation of some fluorescence in the kidney and the liver. These are the clearance organs which you would expect to degrade the nanoparticles and the peptides over time. But the level of fluorescence was significantly less than that seen in the placenta and was transient. The fluorescence did not remain. And we were looking at these tissues up to 72 hours after injection. So once we'd shown that we could use these peptides for placental targeting, we wanted to develop a more biocompatible nanocarrier because we didn't really want to be injecting iron oxide nanoparticles into the placentas of pregnant women. So next slide, please.
Yes.
So we chose liposomes as our biocompatible nanocarrier of choice. There is some animation on here, so I will talk you through the synthesis. We undertook a literature review to find a popular composition that's most widely used in the literature that would give us the parameters that we wanted. We ended up with a formulation of cholesterol and phospholipids, so DSPC and DSPE. We also incorporated some PEG into the liposomes as well, which acted as a stealth layer to prevent aggregation of the particles and recognition and uptake by the immune system. A proportion of the PEG molecules expressed a maleimide group, so that allowed us to covalently couple our placental homing peptides to the surface of the liposomes. We fluorescently labeled them in two different ways. We had a red fluorophore on our homing peptide on the surface of the liposomes, but to begin with, we also incorporated a green carboxyfluorescein cargo into the core of the liposomes. So this way we could track drug release. And these liposomes are about 140 nanometers in diameter and stable for up to a month when kept at 4 degrees C. So next slide, please.
No.
To make sure that the liposomes...
No, please. Linda, please. In the previous slide.
Sorry, I didn't hear that.
The design a biocompatible nanocarrier.
Yeah.
It's very complex to do this.
It's quite easy to do from a practical perspective. The way we would prepare them is we would mix the lipids together, then evaporate the solvent off and prepare a lipid film. We would then rehydrate that lipid film that contained the drug cargo. We would then extrude the liposomes through a filter of a defined pore size. So that helps you to achieve your defined size of the liposome. So we would use a 100 nanometer membrane, and that would allow us to have nanoparticles approximately 100 nanometers in diameter. At that point, we then covalently couple the peptide to the surface, and then we undertake some dialysis to remove any of the unbound peptide and the unincorporated drug cargo. So although it takes 3 or 4 days, it's actually a relatively straightforward procedure to make the liposomes.
Okay, the slide number 10 is in the screen.
Okay, so the targeted delivery.
Yeah.
Okay, so we wanted to then confirm that the targeted liposomes behaved in a similar way to the targeted iron oxide nanoworms. So we repeated our experiments, and it also allowed us to track drug release of our encapsulated carboxyfluoroscene cargo. What I should mention at this point is when encapsulated within the liposomes, the carboxyfluoroscene self-quenches, so you don't see any fluorescence when it is encapsulated. You only see fluorescence when it was released. On the left here you have some images taken of human placenta which have been incubated with liposomes in vitro, and you can see after 24 hours in red we have peptide accumulation, showing that the peptide decorated liposomes had got into that outer trophoblast layer of placental cells, and you can see the corresponding green image to show that the carboxyfluoroscene cargo had been released, so drug delivery had occurred. And in the top left picture there's the yellow colour, which is the overlay of the red and the green. On the right hand side of the slide we have some pictures of the mouse placentas. So with these experiments we injected our targeted liposomes by the tail vein into our pregnant mice. We performed cardiac perfusion, so we removed all the blood from the mice after the incubation time. So that means that any fluorescence we see is from liposome or cargo that's being taken up into the tissue. It's not sitting in the blood spaces. So again, as with the peptides and the nanoparticles, we saw beautiful accumulation in the endothelium of the maternal spiral arteries, those blood vessels that supply the placenta, and also within the placental labyrinth where the nutrient exchange takes place. Next slide, please.
Okay.
So once we validated that the nanocarrier worked, we wanted to then deliver a drug and see if we could improve placental growth and fetal growth. So the aim of the study was to treat the placenta with the aim of rescuing the fetus. So what we did, first we tested it in normal healthy mice. We waited until embryonic day 11.5 of pregnancy, because at this point, this is when the placenta is fully formed and mature and starts to function. Before this point, it is leaky, so we may well have got leakage of our targeted liposomes across the placenta to the fetus anyway. Secondly, if we were to embark on a clinical study, it's usually the middle of pregnancy before you could identify pregnant women presenting with pregnancy complications such as poor fetal growth. So we were trying to mimic the clinical study here as well. We chose to inject 4 times in pregnancy on embryonic day 11.5, 13.5, 15.5 and 17.5, and then we sacrificed the mice on embryonic day 18.5 and measured various parameters including fetal and placental weights. Below this we have the treatment groups. So what we didn't know is what the effects of the empty liposomes would have on our fetal and placental weights. So we had a number of control groups. We had mice that were injected with PBS because the stress, there is a stress associated with a tail vein injection, and stress is a known cause of reduced fetal growth, so we wanted to control for that. We had 3 different types of liposomes: liposomes with no targeting peptide, liposomes decorated with a control peptide that didn't target the placenta, and liposomes decorated with our targeting peptides, IRGD. These 3 sets of liposomes have no drug internalized. We chose to deliver insulin-like growth factor 2 because it's been shown to improve placental growth and placental function. So we had 1 treatment group which was intravenously administered with free IGF-2 at a concentration of 1 milligram per kilogram, and then another 3 treatment groups, each for different liposome formulations, but this time encapsulating IGF-2 at a similar concentration. Next slide, please.
Yes.
So, what you see here is a graph showing placental weights for all the different treatment groups. It's a little bit busy, but what you can take home is that within each litter there is a variety of placental weights. You have smaller placentas and bigger placentas, and actually each mouse doesn't have 1 pup. Each mouse can have between 5 or 10 pups. So within a litter you get a variety of placental found was the median placental weight was significantly increased in the final column of the graph. So that was the targeted liposomes containing IGF-2. And this was compared to our control PBS treated mice, compared to our empty liposomes decorated with the control peptide or the targeting peptide, but also compared to mice treated with liposomes containing IGF-2, but also lacking a targeting peptide or bearing a scrambled control peptide. So we showed that this targeted delivery was significantly increasing placental weight, which we were quite happy by. And if you notice in that right-hand column, you've got lots of heavier placentas, but it's actually the smaller placentas you don't see there. So there's no dots in the lower half of that distribution for that last column of data. And what it appeared to be was that we were making the smallest placentas bigger, but we weren't significantly enhancing the weight of the normal size for bigger placentas. So what we hoped from this point was that we would see a corresponding increase in fetal weight. If you made the placentas bigger, they've got a bigger surface area, so you would hypothesize they could transport more nutrients and you would get heavier fetuses. If we go to the next slide, you've got a graph of fetal weight, and what you can see here is actually there is no significant change in median fetal weight in any of the groups apart from the mice treated with the undecorated empty liposomes, so that's column 2. We were surprised by this initially, but actually what we had found with our fluorescent studies that the plain liposomes lacking any targeting peptide at all did actually cross to the fetus. So when we incorporated a fluorescent cargo in there, we could see evidence of the fluorescent cargo in the fetus. So what we think here in this group is we've just given the fetuses a large dose of lipids, and that's why they've increased in weight. We also hypothesise that perhaps there's regulatory control mechanisms that exist that prevent fetuses getting too big, because it would be detrimental and it would make delivery more difficult. So we think that perhaps there were regulatory mechanisms in play here which stopped our normal fetuses getting too big. So what we wanted to do now was to move on and test our targeted IGF-2 liposomes in a mouse model of poor fetal growth. So where we have fetuses that aren't growing properly, can we make these bigger? So if we could have the next slide, please. So we used a mouse model of fetal growth restriction to study whether we could rescue this poor fetal growth. So the model that we used was the placental specific IGF-2 knockout. So this is a heterozygous mouse, and you get smaller placentas and smaller fetuses. So the way the mice are bred, the mother is wild type and normal, and the father is heterozygous for the IGF-2 knockout. So you get a mixed litter of pups. So the pups are either normal wild type or the P0 knockout, which are comparatively smaller. So on this slide, I plotted fetal weight distribution curves. The weight distribution curves on the left show that the wild type pups, and for this study we used 3 different treatment groups. These mice don't breed particularly well, so this study took quite a long time to undertake. The 3 treatments we used were a PBS control injection, administration of free IGF-2, and a targeted delivery of IGF-2. So what you can see with the wild type pups, those within the mother that were growing normally, there was no significant impact on fetal growth with any of the treatments, which was the same result we saw in the control mice. When we looked at those heterozygous P0 pups, the smaller ones suffering from fetal growth restriction, the blue and the black lines here represent the PBS control and the freely administered IGF-2. Those groups are very similar, but the pink line is the targeted delivery of IGF-2, and you can see we've got a significant shift to the right. We've had a significant impact and improvement of fetal weight, but here again we've appear to make the smaller pups bigger, but we haven't made the larger pups clinically relevant results. When you're treating women in a hospital, you would want to improve the growth of the smallest babies, but you wouldn't want to cause fetal overgrowth of babies that are growing well already. If we could move on to the next slide, please.
Yes.
We have carried out a parallel study. We wanted to see whether we could use this time a vasodilator delivered to pregnant mice using a different targeting peptide to see if we could increase fetal growth. So rather than give a growth signal to the placenta, this time we give a vasodilator to the uterine vasculature. And we identified a molecule called SE175, which is a nitroxyacetylated thiosalicylate. It's quite an interesting molecule. It's stable in buffers in the laboratory, but as soon as it comes into contact with cells and tissues, naturally occurring esterases will hydrolyze this molecule, so you get a nitrate limited and a thiosalicylate limited. And the three-phyl group of the thiosalicylate can then liberate nitric oxide from the nitrate group, so you get spontaneous generation of nitric oxide, which is a known vasodilator. Even more useful is that thiosalicylate that's generated can act as a cyclooxygenase inhibitor. And actually, cyclooxygenase is upregulated in some placentas from pregnancy complications. So inhibiting cyclooxygenase may also reduce some of the inflammation and oxidative stress that's observed in some pregnancy complications. So the graph at the bottom of this slide just shows the ability of this compound SE175 to relax mouse uterine arteries. So this is an in vitro experiment. We've got pieces of pregnant mouse uterine artery and we've applied SE175. This is the black line on the graph. And compared to a vehicle control or a time control, we saw significant relaxation of these pre-constricted vessels. So this showed us that this in vitro, this compound was an excellent vasodilator. So at this point, we then went to try and encapsulate it into our liposomes to see whether it would work in vivo as well. Next slide, please.
Yes.
So we undertook a very similar treatment study to that with the IGF-2, injecting 4 times during pregnancy and then sacrificing on E18.5. Again, we reduced a number of treatment groups here. We used a vehicle control, so PBS injection. We used a different peptide, so this NKG sequence we showed targets the spiral arteries of the placentas, so this is more of a vascular targeting peptide rather than targeting the labyrinth and nutrient exchange area. So we used liposomes containing PBS and decorated with this NKG peptide. We used liposomes containing the SE175, decorated with this vascular targeting peptide. And we used free SE175 administration as a control. Next slide, please.
Yes.
So here we used healthy mice again, and we saw very similar results to when we used IGF-2 as a drug of choice. So in healthy C57 mice, we didn't see any significant change in fetal or placental weights in our mice. We were slightly disappointed, but we weren't surprised at this point. So in parallel, we were carrying on studies with an endothelial nitric oxide synthase knockout mouse. So this is characterized by high blood pressure, and in pregnancy it maintains that high blood pressure. You also see protein in the urine and some changes to the kidneys. So it's a very good model for pregnancy complication and preeclampsia. So we can have the next slide, please.
Yes.
So using this mouse model of preeclampsia, here on the left we have a graph of fetal weights, and we saw a significant increase in fetal weights when we used targeted delivery of the SE175 compound compared to the PBS controls. But we saw no effect with the freely administered SE175. On the right-hand side, we plotted out the fetal weight distribution curves, and we marked on the 5th and 10th weight centile, so that's the smallest mice. So what you see is there's the blue curve is the control, and the pink curve is the free SE175. So those curves overlap. Free SE175 had very little effect on fetal weight distribution. In red, we have the PBS, loaded targeted liposomes, where the curve is shifted slightly to the left, so actually those fetuses got slightly smaller. But in green, we have our targeted delivery of SE175 to the uterine vasculature, and you see a slight shift to the right of that curve compared to the control. And in that group, again, there were no fetuses that fell below the 5th or the 10th weight centiles. So similar to what we saw with our mouse model of fetal growth restriction, we seem to be making the smallest fetuses bigger, but not making the normal and big fetuses any bigger. Again, a clinically relevant outcome. Next slide, please.
Yes.
So we were also interested in the effect on placental weight. So the vasodilator shouldn't have acted directly on the placenta, it should just be acting on the uterine blood vessels. And what we actually saw was a significant decrease in placental weight in both mice treated with the targeted SE175, but also the free SE175. The ENOS knockout mice are characterized normally by a slightly larger placenta than normal, and this is reported to be an adaptation to the reduced blood supply and the high blood pressure. So what we've actually done by seeing a reduction in placental weight, we believe we've normalized placental weight. By giving a vasodilator, we've improved blood flow. There was less stress on those placentas, so there was no need for them to adapt and become bigger. We also measure fetal to placental weight ratio, which is just fetal weight divided by placental weight. So that tells you how many grams of fetus can be made by a gram of placenta. So what this shows is placental efficiency, essentially. A smaller placenta making a bigger fetus is working more efficiently. So both the targeted SE175 and the free SE175 have acted here to improve placental efficiency. So we've seen decreased placental weight, but corresponding increase in fetal weight. So this is really exciting for us that we can deliver a drug to the blood vessels of the uterus and see alteration of placental weight as well as fetal weight. This is currently unpublished. We are currently writing this up for submission. So just to summarise this part of the talk, now we've got data using 2 different targeting peptides, 2 different drugs, and in both healthy mouse, mice, and 2 different mouse models of pregnancy complications, we've shown that these targeted liposomes can be used to selectively deliver drugs to the placenta and the uterine vasculature and see clinically relevant enhancements of fetal growth. The next slide, please.
Yes.
I'm just going to finish the presentation by briefly talking about a different project that we've been working on, trying to manipulate intrinsic placental growth signaling. So rather than try and deliver an exogenous growth signal or an exogenous drug that might have off-target effects in different tissues, we wondered whether we could manipulate intrinsic or growth signaling within the placenta. And one of my colleagues was interested in microRNAs and their ability to regulate placental growth. Now these are small non-coding RNA sequences that inhibit protein translation. And we wondered whether we could alter microRNA expression within the placenta. There's specific microRNAs that are linked to control of placental cell growth. Then we could potentially manipulate placental cell growth in a slightly different way. It's been reported that microRNA 145 can suppress IGF-1-induced trophoblast proliferation in human placenta, and also microRNA 675 is really important for mouse placental growth and suppresses growth and IGF-1 receptor expression. So both MIR145 and MIR675 are negative regulators of growth. So if we could inhibit those negative regulators, the outcome should be enhanced placental growth. So our hypothesis was to develop some microRNA inhibitors we were going to couple our homing peptide sequences to those microRNA inhibitor conjugates. Next slide, please.
Yes.
We couple a peptide sequence to a microRNA inhibitor, which was typically made out of RNA. And the way we went about this was to, instead of using an RNA-based inhibitor, was to synthesize our microRNA inhibitors from peptide nucleic acids, or PNAs. So these are very similar to DNA or RNA, but the ribose backbone, which you would see in DNA or RNA, is replaced by a modified glycine backbone. So I've got some images on this slide of the structure. So you see a thicker backbone, but you get the base pairing as you would in DNA or RNA. And actually these are very stable in vivo because there's no naturally occurring proteases that can cleave them. So they make excellent small molecule inhibitors. We ordered these from a commercial company, and we got 3 different peptide nucleic acid synthesized. The first one was a scrambled microRNA inhibitor linked to a targeting peptide, and this time we used the CGKRK sequence. We also got a MIR145 inhibitor linked to a CGKRK homing peptide, and a MIR675 inhibitor linked to the CGKRK homing peptide. Next slide, please.
Yes.
So, as previously, we did some experiments in wild-type mice. What you have here, the top 2 graphs, are looking at placental weight following treatment with the different microRNA inhibitors. Again, we've used an injection of PBS as a vehicle control. What you'll see in the first graph on the top left is that compared to both the PBS and the scrambled inhibitor conjugate, the targeted MIR675 inhibitor significantly increased placental weight compared to control. So that's median placental weight shown here. And if you plot the placental weight distribution curves as shown on the right, the black line and the blue line that overlay each other are the PBS control and the scrambled inhibitor. So the scrambled inhibitor is having no effect on placental weight. But in the red and the grey, you have the MIR145 inhibitor and the MIR675 inhibitor. And again, you get that shift of the placental weight growth, the placental weight curve to the right. So you've got fewer smaller placentas, but you haven't got any change in the number of bigger placentas. The graphs below show fetal weights. So we also saw a significant increase in the median fetal weight, both with the MIR675 targeted inhibitor and the MIR145 targeted inhibitor, which we were very pleased to see. What we were interested in also to see, looking at the fetal weight distribution curves, is we plot these. Although we didn't see a significant increase in the median fetal weight, the weight distribution curve for the scrambled inhibitor also looks very similar to that for 2 targeted inhibitors, the MIR145 and the MIR675 inhibitor. So there's some effect there of our scrambled inhibitor on fetal weight, which we need to look into a little bit more. But again, this is evidence of another promising approach, this time inhibiting intrinsic growth signaling using microRNA inhibitors to alter fetal and placental weights. Next slide, please.
Yes.
So just to summarize for you, I hope I showed you some different ways in which we've created some different nanoscale therapeutics, either liposomes decorated with targeting peptides and containing a drug, or microRNA inhibitors coupled to targeting peptides. And our ongoing work is to test these different drugs and different peptides and different mouse models of pregnancy complications. And we aim to improve some of the clinically relevant outputs you might see. So we would want to increase fetal weight and placental weight, or enhance placental function, and enhance uterine blood flow, and reduce some of the side effects you see in the offspring associated with pregnancy complications. Next slide, please.
Yes.
So I'd just like to finish by acknowledging the large number of people that have been involved in this work. I've had 2 excellent PhD students, Annie and Ashley Curitan, at the University of Leeds, helped with the microRNA work, and then Professor Russ Larty at Stanford Burnham Medical Research Institute, some of his staff were very helpful with the initial screening and have provided support and guidance along the way. The majority of this work was funded by a David Phillips Research Fellowship from the BBS, so that's the to myself, and we've had some other funding from Thomas the Baby Charity and the Nano PhD scheme. So thank you very much for your interest.
Okay, Linda, thank you for your presentation. I would like to tell you that is the first time that I see and now I know something about the nanotechnology and the placenta problems that nanotechnology can contribute to solve some problems. So I will asking for some questions for you. Maybe is for you, maybe is stupid question, but but is if you understand in general, you use nanoparticles because it's more efficient than macroparticles. Is this?
It is true. I think what we have done is copied strategies that have been used successfully for cancer and cancer targeting. And there's a lot of work showing that liposomes are effective at this particular size range, and that's why we've taken this approach. Secondly, there is a liposome formulation that's already licensed for use in humans for delivering chemotherapeutic drugs to tumours. So Doxil is an FDA licensed formulation as a chemotherapeutic drug, doxorubicin. So we thought if we could follow a similar protocol, similar formulation that's already been licensed for use in humans, that would give us the greater likelihood to get this work translated clinically into humans. It may well be that macroparticles might be more efficient, and that's something we'd certainly be interested in looking into. But at the moment, we just focused on liposomes based on previous literature.
And the other question is about this kind of nanoparticle could goes to the body of the mother of the children.
So the non-targeted nanoparticles, do you mean?
Yeah.
Yeah, so what we've seen so far is if we use liposomes that don't detect, because they don't express the targeting peptides, these will cross to the fetus and they will accumulate much more in some of the other maternal organs, such as the lung, and you see a lot more in the liver and the kidney and the spleen compared to the targeted nanoparticles. One thing we're interested in is whether we can identify peptides that selectively target the fetus, because we might be in a situation where you can detect fetal abnormalities in utero, and you might want to be able to deliver drugs through the placenta to the fetus to maybe correct a developmental defect. So potentially we are interested in the properties of liposomes that allow them to cross the placenta to the fetus. But in our current studies, we would want to reduce the risk of any adverse side effects by limiting placental targeting just to the placenta and trying to prevent those particles crossing to the fetus.
In this kind of research, you can substitute the animals or the mice for computer simulation.
I think we could. There's has been work by a number of groups simulating the placenta and placental blood vessels, and there's some really good mathematics now to model exchange within the placenta. So yes, potentially that would be really useful for us. That could tell us ways of optimizing size or surface charge, ways of understanding transfer of substances from the maternal circulation to the fecal circulation and changes in blood pressure and secretion of factors across the placenta. So again, it would be really interesting to see what happened to our liposomes if we studied them in this ex vivo model of the human placenta.
And this is more expensive or more cheap? This process, the computer simulation, is more expensive or more cheap for the research?
I think it would be cheaper, definitely. You can't argue with that. So yes, this would be a really interesting and cheap way to perhaps advance this research.
Okay, and in general, to do this kind of research, how much money is you need?
My initial fellowship to undertake this work was for 5 years and was for about £600,000. So I guess that's about $1 million US. It's because we were starting from scratch, because there was nobody else in the literature who'd done anything similar. We spent a lot of time doing testing a lot of different formulations, running a lot of control groups and using a lot of animal models. So actually the initial workup was quite expensive. But now we know we've got a formulation that works, we've got peptides that we know work, and we understand a bit more about what happens to the control groups. It's going to be significantly cheaper from now on in planning more mouse experiments. But when you work with mice and when you need 8 to 10 mice per treatment group, you're getting through quite a lot of animals. So there is a significant cost associated with this kind of research.
And you use a public fund or private fund?
Publicly funded, so by the UK government, they funded, the BBSRC is a UK government funded research agency, and they funded my fellowship to do this work.
Yeah, because I don't know in England, because here in Brazil, all of research for nanotechnology is with public fund. I don't know in England if one private company are interested in put money to do this research.
I think there are some pharmaceutical companies that are interested in some nano formulations. There's definitely improvements to be made in the way drugs can be formulated and developed by packaging nanoparticles. So I think there are small pots of money available, but I think you're more reliant on the pharmaceutical companies being aware of your work and contacting you. I think it's harder for you to go to them and ask for support. So I think at the moment a lot of the research relies on government-funded projects.
And the humans that have problems with the placenta contribute for your resource.
So do they donate their placentas, do you mean? Or do they is there money from charities? So you can, it is easy if you want to get the tissue from women with pregnancy complications. They're usually very happy to donate their placentas for research, so there isn't a shortage. They're also very happy to contribute to research studies and be involved. And there are also charities within the UK that support women's health issues, so problems in pregnancy, and charities that support poor fetal growth. So there are some small charities that support this kind of research as well, but it's much less well funded than things like heart disease or cancer.
Okay. Lisa, thank you so much for your contribution with our program. I think this is a very important resource, and is for us the first time that he have some knowledge about this relationship between nanotechnology and this kind of the problem for the humans. No?
Yeah, well, thank you very much for being interested in my work and giving me this opportunity to tell you about it.
Okay, see you. Bye bye.